Good morning, everyone. Welcome back to High School Students Conference on Inquiry on Practice in Relation to Sustainable Development Goals. The fourth presentation session is about to begin. Please be seated. To all presentation teams, please note that each team has 12 minutes to present. A bell will chime 1 minute before your time is up. When you see the sign reading time is up, please wrap up your presentation within 30 seconds to ensure a smooth flow through the agenda. And now, let's have a brief introduction of professors and commentators on the fourth section. Professor Cai Qinli from NCKU. Professor Xie Zhongling from NTU. Professor Wang Haowen from NCKU. Professor Liu Guangyan from NCKU. And of course, we got our Dr. Liao Taigu, Principal Liao from National China First Senior High. So this session is about environmental protection. 4 teams will present to us their innovative ideas. The first presentation is micropaper Daniell cell applicated in chemical analysis, presented by National Pingtung Senior High School. Please welcome them with a round of applause.
Good morning, everyone. We are students from National Pingtung Senior High School. Our topic is micropaper Daniell cell applicated in chemical analysis. I'm James, and here are our group members. They are Alan, Leo, and Channing. First, let's look at our motivation and objectives. When we do the chromatography, those multiple samples might overlap together, and it will cause us difficult to distinguish and resulting in poor separation efficiency. Therefore, we drip solution on the filter paper to replace the beaker and U-shaped pipe. By doing so, we can reduce the volume of solution. To deal with this challenge we have found out, we try to discover a new kind of Daniell cell, which is called micropaper Daniell cell. First, let's look at this. This is a traditional Daniell cell device. In this experiment, we need 2 cups of solution and 1 U-shaped pipe. However, this needs a lot of chemical waste. In this part, you can see how we conduct our experiment. First, we design the device by two circular filter paper and 1 strip of filter paper to replace the 2 beakers and a U-shaped pipe. Second, we drip electrolyte solution on both the circular filter paper and the strip of filter paper. Next, we put metal sheets on the circular filter paper as the electrodes. And the last step, we measure the electric voltages and currents of micropaper Daniell cells. And you can look at the schema. Next, I'll invite Alan to talk about this further.
Okay, in part 1 experiment, we focused on optimize the micropaper Daniell cell and output the maximum electric current. We decide 4 independent variables, and I will talk them in detail later. Independent variable 1, type of salt in a salt bridge. We control these variables before we perform each experiment. And according to the results, We find potassium chloride produces a maximum electric current and voltage. It produces 478 microampere. Independent variable 2, types of health cells paired up. We use 3 kinds of metal sheet in these experiments. They are copper, zinc, and aluminum. Through the results, we can know zinc health cell paired up, copper health cell produces the maximum current and voltage. And furthermore, we also find zinc health cell paired up, aluminum health cell produces extremely low current and voltage. We assume that it is because their activeness is too close. Independent variable 3, concentrations of the solution in the health cells. We prepare several different concentrations of zinc and copper sulfate solution. Pairing up 2 health cells, we could get 16 different combinations. According to the results, coupling 2N zinc ion in zinc health cell with copper ion in copper health cell produces the maximum current. And independent variables 4, size of metal sheet as cell electrodes. We use 3 kinds of square-shaped metal sheets in the experiments. They are 4, 16, 13, 6 square centimeter. According to the results, we can get 13.6 square centimeter of square-shaped metal sheet produce maximum current. Among the experiments we did, we speculated, first, different substances might present different movements. And the second, zinc health cell with aluminum health cells has extremely low current. We predicted that it is because their activeness is too close. Third, 2N solution will produce a maximum current because their higher concentrations will increase the collision probability. And the next, under our experiment design, 13.6 square centimeter of square-shaped metal sheet will produce the maximum current. It's related to the high contact area between the field paper and metal sheet. The last, with just a little solution in the salt bridge and metal sheets, the device actually complies with the principle of green chemistry, prevent waste. Then we make sure use of this device with the optimum conditions to develop a new analysis method, which will be introduced part 2.
And next, I'm going to talk about the part 2 of our experiment. First, because of the different molecule weights and the shapes and dissociation degrees, we want to take advantage of the above features to analyze and identify the substances by our micropaper Daniell cells. And next, this is the structure of tazrazine and nucleoxine. In this part, first, we assume these 2 compounds will dissolving in water into the sodium cations and the corresponding anions. And we also assume the generated anions with a specific color will extend toward a cation. And next, this is the schema of part 2. First, to help us observe the movement of the currents, we change the distance between the 2 circular filter papers to 2.5 centimeters. And then we use a capillary tube to spot a small amount of the pigment onto the middle of the sheet of filter paper. And then we connect the 2 health cells with a wire and observe the movement of the currents. And this is the result of part 2. First, this is the tazrazine movement. And you can scan the QR code on the button and you will see the movement of tazrazine. And in this part, the yellow anions will extend at the rate of 0.102 millimeters per second. And next, this is the movement of nucleoxine. And in this part, the nucleoxine will extend at the rate of 0.081 millimeters per second. And this is as we predict. The yellow anions will move quicker than the red anions. And we know that the anions of pigment compounds with specific color will move toward the zinc health cell and show a viewable track on sheet of filter paper without extra internal electricity. And we demonstrate that every ions will move or extend in different speeds because of the different substance. And next, my teammate Channing will talk about the conclusion.
And okay, I'm going to talk about our first conclusion. And our experiment is compliant with the SDG 6.3, improving water quality through measures such as reducing pollutions. Since we use the filter paper to replace the beaker and the U-shaped pipe, we only use a small amount of solution. And then, eliminating waste dumping and minimize the release of hazardous chemicals and materials, aiming to have the proportion of untreated wastewater. Although the zinc sulfate solution and the copper sulfate solution is a toxic solution, but after a well recycled, and they won't pose a big threat to our environment. And next, our experiment also complies with the SDG 9.5, enhance scientific research across all nations and elevates technical capability in industrial sector. In our experiment, the Daniell cells has really a potential to analyze an unknown substance. And best of all, it did not have the external electricity. And then this complies with the two principles of green chemistry, prevent waste and design for energy efficiency. And in the future, we hope that this kind of device can be used in the education aspect and let many students know that not only chromatography, but also our Daniell cells can, analyze the unknown substances. And then, best of all, we hope that this kind of device can be used in research aspect and for the scientists and researchers who face the same problem when the chromatography doesn't work, and they can take advantage of this device as an alternative. And next, this is all our references. All our picture and our information are come from here. And thanks for your attention.
Thank you, Pingtung Senior High School, for your presentation.
Good morning, Principal Liao. Well done. Okay, another day and another set of good presentations. Okay, I'm going to comment on the first one, the Daniell cell applications from the Pingtung High School, okay, Senior High School. I'm particularly impressed about the multi-layered of the research project. Where are you? From Pingtung? Yes, because initially I thought you were doing the battery study, okay, just designing Daniell cells. But as the presentation went, okay, I found that the Daniell cell is just a tool because you use a Daniell cell to really try to separate substance, okay, and try to mimic the functionality of the chromatography applications. So I think that's a very genius design because, as you mentioned, that it fits the SDG goals, okay, of 6.3 and 9, okay, in terms of reducing the waste, okay, the material waste, the water waste, and also you have industrial innovations, okay, on your project, okay. And so you started with, studying the parameters of batteries, try to optimize the batteries, and then you use a salt bridge as a media, okay, diffusional media, okay, and then you can separate different chemicals, okay, based on, although you use just like a basic, like a pigment or like a color chemicals and try to demonstrate the idea. But I think it is a very simple way to showcase a very complicated idea, okay. However, the diffusional behaviors of your salt bridge, I think still can be improved because if you study closely, the mass transfer of materials, okay, depends on many factors. For example, you might be able to tilt two electrodes and the gravity will help you to move the materials, okay? So in terms of mass transport, not only the diffusional diffusivity, but also you can also consider other sort of mechanism that help you to move the material. For example, gravity or some kind of streaming, okay, design in order to separate the substance further, okay? But as I said, it is a very, very smart experiments, and I like the systematic nature. Okay, you study the parameters one by one. Okay, including the the optimization of the batteries. Okay, in terms of the pairing of electrodes, the size of your metals. Okay, metal plates. Okay, and the chemical, the electrolytes you use, okay? And I think there's one more aspect that you might want to look into is how long, how steady the current, the voltage can last, okay? Because you only use few, you just weight the papers basically, and how long the longevity, the lifetime of your current, of your voltage, the steadiness, okay, is the key to have a good separation. Okay, and how long can your device last? I think you can bring the lifetime parameter into your study as well. But overall, I think it's a perfect experiment, multi-layer experiments, okay, to demonstrate the ideas of your SDG goals. Okay, thank you.